students. In this video, we will start with Chapter 7, Section 2. In Chapter 7, Section 2, we will review together what we mean by phenotype and genotype first, and then after that, we will go to talk about probability, monohybrid, and dihybrid cross. First of all, we said before that genotype is the genetic makeup of the organism, and usually it consists of factors or alleles, and it's represented by letters. For example, when we say P small, A capital, A capital, A capital, A small, or T small, all of these are letters. So when you see letters, this is the genotype of the organism. And for example, or maybe, for example, he say heterozygous, and you know that hetero, for example, this is hetero, or homo, for example, like homo, which is named as pure or true breeding, as Mendel said before. These homo, for example, you have P capital, or P small. All of these are the genotype of the organism. Okay. The genotype, we have two types of genotypes. As we said before, we have homo, which is mean it's pure. And when we say homo or pure, also we can say true breeding. These homo or pure, we have two types. It's either homo dominant. When it's dominant, it should be capital letter, capital letter. This is represent the homo dominant. And the homo recessive, it's always a small letter, small letter. What about hetero? Hetero, it's only for the dominant trait. The other name for hetero, it's hybrid. When we say hybrid or hetero, it means you have, for example, uppercase and lowercase of the letter. For example, we have A capital, A small. This represents hetero, and it's always dominant. We don't have recessive for it. So very important, if the alleles or pair of alleles are alike, they are similar to each other, like here, A capital, or A small, it's named as pure or homozygous. But if the both alleles, they are different, they are not alike each other, like here, A capital, A small, so we say it's hetero or hybrid. Okay. So when we say homo, hetero, hybrid, pure, all of these are representing the genotype. What about phenotype? Pheno, it means the appearance of the organism. It's the organism appearance. For example, you say tall, you say short, you say purple flower, you say white flower. All of these are appearance of the organism, and it's represented by words. It's not by letter. For example, they say purple flower. This is a phenotype of the organism. Or inflated pod. This is another phenotype of the pea plant. So the phenotype, for example, for P capital, or P capital, P small, it's purple. We say it's purple flower. And the phenotype of P small, it's what? It's white flower. Okay, this is the phenotype. So the difference between geno and pheno: geno by letters, pheno by words. Geno represent by letters and represent the genetic makeup of organism, while the phenotype, it's the appearance of the organism. If I talk about the phenotype of the dominant specifically, for example, I say Yara go to bring purple flower. When I say purple flower, which is a phenotype, you say which one, teacher? Is it P capital, P small, or P capital, P capital? So you don't know from this phenotype that I said, which is purple, you can determine if it's P capital, or P capital, P small. Why? Because if the recessive factor come with the dominant factor, at that time it also appear by the dominant trait. So this is the first reason, because of the recessive alleles. For example, when I say purple flower, you don't know if it's P capital, or P capital, P small, because when the recessive factor come with the dominant factor, it's also appear by the dominant. Okay, so we say the phenotype does not always represent genotype. The second reason is because of the environmental factor. The environmental factor also can affect, because the genotype may not reach its full potential. What do you mean by that? Due to the environmental factor. What do you mean by that? For example, you have plant that usually it's tall. You get tall seeds to grow the plant, which are tall. But sometimes, because of the lack of nutrient in the soil, lack of water, or anything else, or lack of, for example, not enough sun exposure, so these plants that should be tall in their genotype, they might grow into short. So this is an environmental factor that can affect what can affect also the phenotype. And that time, this phenotype does not always represent the genotype. This is only in Mendelian genetic, okay? Not in the other genetic, like we will talk about them later, incomplete and codominance. What about if I say the genotype represent phenotype? can you know the phenotype from the genotype? Yes, this is always it's correct for all types of Mendelian and non-Mendelian genetic. Because if I give you the genotype, for example, I say it's P capital P small, you already will know it's for purple flower. Okay, and if I said, for example, R capital, you will directly know that the phenotype it's for round seed. Okay, if I say, for example, R small, you already will know that it's for wrinkled seed. So from genotype, yes, you can always indicate the phenotype, but from pheno, we can't always determine the geno. Which one? For which one? For Mendelian genetic, which means for complete dominance. Now let's start with other subject, which is named as the probability. The probability, what do you mean by probability? You expect something to happen. So it's the likelihood that a specific event will occur. So you expect that something will happen. For example, the probability of your success in the exam, you say, I expect that 50% I will pass the exam with a high degree. So this is, you put a probability. So probability, what we mean by probability? It's the likelihood that a specific event will occur. You expect something to happen, and this expectation or the probability may be expressed as decimal. For example, you have when you have 25%, it's all 25 or 50 or 75, or maybe 100 or 100%, 10. So, or maybe as a percentage, for example, 25, 50, this is 75 or 100. Or maybe represented as fraction, and you know what is fraction in mathematics, or maybe as ratio. What about if it's, for example, we have also 1 1 1 0, 0 1. Okay, and for a ratio also we have, for example, 1 1. The most questions in the ministry come as ratio. For example, we say the ratio is 1 1. What do you mean by 1 1? It means 50 50. Or half of them, for example, tall and half of them short. We have 1 3 3 1, 1 2 2 2 1, and 9 3 3 1. We have many different ratios. We will talk about each one in detail later. And monohybrid and dihybrid cross. Okay, now what is the rule or the equation for measuring the probability? The probability, we have this equation, and you should memorize this equation, is the number of times an event is expected to happen, very important, okay, this one that you expect to happen, on the number of times an event could happen, which means the total number, okay? So in the beginning, you have, for example, the question asked about the probability of the dominant. So the dominant, it's unknown. You expected how much to happen, okay? We don't know how the dominant will appear. And we divide it by what? By the number of times the event could happen, which means the total number. Let's take an example to know how we can solve this type of question. So the probability is the number of time an event expected to happen on the total number, or the number of time the event could happen, which means the total here. So we can say part on the total. Now let's take an example for it. In Mendel's experiment, the dominant trait of yellow seed, you know that the seed color, the yellow is dominant and the green is recessive. This is the dominant and this is the recessive. So the yellow seed appeared in F2 generation by this number, and the recessive by this number. What about if the question asked about the probability of the dominant? So at that time, we will put the part on the total. Which part he want? He want the dominant. The dominant, how many, the number of dominant is 6,000 or 0.222. So we will divide it on what? Divide it on the total number. Please pay attention, it's on the total. So first you should take the summation of these two together and then divide the dominant over the total. What about the probability of recessive? At that time, we will put the number of the recessive on the number of the total of both of them. What about if he asks about the ratio of the dominant to recessive? The dominant is 0.75 over the ratio of recessive is 0.25. So it's like 3 to 1, dominant to recessive. Sometimes he asks you about the number directly, the number of time an event could happen. For example, the number of dominant. At that time, you can multiply these two, the probability that you have by the number of time that an event could happen, which means the total. So multiply the probability with the total, you can get what? You can get the dominant or the recessive trait. For example, he asked about the number of dominant. He gave you that the ratio that it's appear, or the probability that it appear, it's 3 over 16. You multiply it by what? You multiply it by the total number. For example, we have 600. At that time, the dominant will appear. The same thing when you have a coin. For the coin, you have two sides, so you have head and tail. Whenever, every time you throw the coin, it's maybe 50% appear with head and 50% appear with tail. And when you, as you make more and more of the trials, at that time you can get this ratio, which is 50% in each time that head will appear or tail will appear. What you mean by genotypic ratio and phenotypic ratio that most questions ask about it is you put the letters, for example, in the ratio, the offspring that you get. For example, if you cross a capital A small with a capital A small, at that time what will appear is that you have one of them will be, for example, a capital, two of them will be a capital A small, and one of them will be a small. So this is the genotypic ratio. You should focus on the different type of letters that you have and put them in a ratio. Or we can say 25% a capital, 50% a capital A small, and 25% a small. This is for the genotypic ratio. You should focus on the letters only. And for the phenotypic ratio, you should focus on the appearance of the organism. Okay, for example, we say purple, we say white. For example, we can say 3 purple to 1 white, 3 to 1. This is a phenotypic ratio. Now, what do you mean by Punnett square? Before we study the monohybrid and dihybrid cross, we should know what you mean by Punnett square. Punnett square is the square that the biologists use for what? Is it for predicting for example, the parent? It's for predicting the probable distribution of what? Of inherited traits in offspring. So it's for the offspring. We will put the offspring here, and we will see the distribution of the offspring and the inherited traits in the offspring. This is the Punnett square. Okay, we will put the gametes above here, and then we'll start crossing them to get the offspring. Always the recessive, don't forget that always recessive represented by two letters with small letters, okay? And they are always homozygous. What about the dominant trait? The dominant may be hetero or hybrid, like for example T capital T small, or maybe two capital letters. So the dominant trait, we have two genotype for it. Maybe capital letter with capital letter or capital with a small. If it's capital with capital, this is homo or pure. If it's capital with small, it's named as hetero or hybrid. Now let's go take an example and solve this question to see what we mean by Punnett square. In a cross between two heterozygous parents. So first of all, you should translate that. What do you mean by heterozygous parent? For example, take an example of any letter that you want. This is hetero. And he said that we have two hetero parent. This is two hetero parent. If we cross these two hetero parent, the resulting generation, he said, they are 400 offspring. So let's do it in a Punnett square. This is a Punnett square. And we will put the gametes here. For example, we have a capital A small and we have a capital A small. And the result of it, when we will put these gametes here. And these gametes, how we get these gametes? By the law of segregation, because in Mendel's first law, we said that it's for monohybrid cross, which means when we have one trait, one character. For example, here we have a capital A small and a capital A small. So according to law of segregation, these two factors or allele will separate from each other during the formation of gamete. And during the formation of offspring or the zygote, they recombine again with each other. So here we have a capital, then we have a capital A small, then we have again a capital A small, and then a small a small. Okay, this is the Punnett square. So Punnett square is used for what? It's used for this for predicting the probable distribution to distribute what? The inherent rates of the offspring. Here we can see the offspring here. So we can say here in the question he said they are 400 offspring are produced. So we can say this one is 100, this one is 100, and each one from these is 100. So what is the expected number of resulting heterozygous individual? How many of these individuals are heterozygous? Okay, let's change the color. We have this one, it's hetero, and this one is hetero. So 200 of them are heterozygous. What about if I asked about homodominant? Homodominant, it means this one, A capital, A capital. So it's only 100. Okay. What about if I asked about homorecessive? This is homorecessive is also 100. What about if I asked about homo? What is the number of homo? This is homo and this is homo. So it also will be 200. Now, what do you mean by monohybrid cross? In monohybrid cross, we will focus on only one character, okay? For example, when I talk about the height of the plant, I have tall and short. When I talk about the flower color, we have purple and white. So we focus on one character only. In monohybrid cross, it's a cross in which only one character is tracked. This is the definition of monohybrid cross. This is a monohybrid cross, for example. We have many cases for it. We will study each case and we will know how to solve the questions of monohybrid cross. It's very easy. For example, in case 1, if we cross between a tall homo pea plant with a short homo pea plant. So we have two homo tall, which means homo dominant, and the other one is short, which means homo recessive. So if you cross homo dominant and homo recessive, like for example, we have T capital, which is tall, with a recessive, which is T small. At that time, what is the predicted ratio of the offspring? This is the Punnett square, and for example, we will put the gametes here. This is for the first parent, which is T small. Based on the law of segregation, the first law of Mendel, these gametes will separate, the letters or the alleles are separate during the formation of gamete. And this is the second parent, for example, we have T capital. Again, it will separate during the formation of gamete. So these represent the gametes here. Then we will recombine them together in order to make the offspring. This is the first offspring, and from this and this we will get this one, and these also, all of these offspring, you can see they are what they are, T capital T small. So all these offspring, are they tall or short? They are all tall. As phenotype, we can say all of them are tall, and as genotype, we can say all of them are T capital T small, or all of them are what are heterozygous. So whenever you cross homodominant with homorecessive, you will get what? We will get all of them are tall and all of them are heterozygous. Maybe in the question, he said that you have unknown parents. For example, you have these unknown parents, and all the offspring appear with dominant trait and they are hetero. So at that time, you know that one of them will be homodominant and the other one will be homorecessive, because 100%, if it's appear dominant and hetero, so it means one of them will be homodominant and the other one will be homorecessive. You can do it by this Punnett square. You can memorize it. Whenever you have homodominant with homorecessive, all the offspring will be 100% heterozygous, which is dominant. Or you can easily make this by the crossing this by multiplication. You will omit the repeated one, and at that time you say that all the offspring will be what? Will be T capital T small. This is the easiest way. Okay, do you remember in the experiment of Mendel, which step is it for making the P generation, the F1 or the F2? The result is all of them will are heterozygous and 100% dominant. This is like the F1 generation of Mendel. He get F1 how? By crossing two parents, and these two parents are homo. One of them is homodominant and the other is homorecessive. Or we can say two contrasting traits, or two true breeding. All of these are correct, okay? Or two pure parent. When you cross two pure parents here, or two contrasting trait, or two true breeding here, they are contrasting. At that time, you will get 100%. They are all heterozygous and they are all dominant. So like the F1 generation of Mendel. So this is in case 1. If you have homodominant and homorecessive, we said all the offspring will be hetero, 100% tall. So if we talk about the genotypic ratio and phenotypic ratio, we said in phenotypic ratio, you should focus on the appearance. The appearance of this organism, is it tall or short? It's all of them are tall. So we say 100% it's tall, or we can say 10. It means 100% all of them are tall and 0% it's short appear. So it means all of them are tall. What about the genotypic ratio? We focus on the letters. The letters that appear, as you can see here, all of them are with the same thing. So we get 4 of them, 4 offspring, all of them are T capital T small. Instead of writing 4, we say 100% T capital T small, or 1 to 0, or 4 to 0, which mean all of them are tall and 0% short. This is for the genotype. We focus on the T capital T small. We can't say, sorry, tall or short. We say only all of them are T capital T small, which is heterozygous. So this is the genotypic ratio, 1, 0. It means 100% all of them are T capital T small and no other type. In case 2, we will focus on what we will cross heterozygous, like for example, R capital R small, which is for round seed, with homozygous dominant. So whenever you cross hetero, like here, we have R capital R small, which is for round seed, with homozygous dominant, R capital R capital. So half of them will be with this parent and half of them like this parent. So we can say 50% will appear R capital, and 50% R capital R small. But what about, this is for what? This is for the genotype. We can get two different genotype. This type of genotype, which is R capital, so two of them are R capital, and two of them, as you can see, are R capital R small. How we get them? By making the Punnett square for them. We will put the first parent gametes here and the second parent gamete here. Then we will multiply them and we get these two type. So we have two genotype, two different genotype, or you can say 1 to 1. The half of them are R capital, and half of them are capital R small. Half of them are homozygous and half of them are heterozygous. What about the phenotypic ratio? This is round, this is round, this is round, and this is round. So you can say 100% they are round seed, or 1, 0, which means all of them, or 100% they are round seed, and there is no wrinkle seed or no other type. Now let's take another example. If we cross hetero with homorecessive instead of homodominant. If you cross hetero, like for example a capital A small, with a homorecessive, like A small. By the way, recessive it's always homo, so if we omit this, it's also correct. We can say hetero with recessive. Hetero with recessive is an example of test cross. We will talk about it later. So if you have a capital A small like hetero with recessive like A small, at that time what you can get half of the offspring are hetero and half of the offspring are homorecessive. So if you cross heterozygous, like for example you have a capital A small with another parent which is homorecessive or recessive trait, for example, at that time in the Punnett square you can see that you have half of them will be a capital I small and half of them will be a small. So we can say like that 50% will be a capital A small and 50% will be a small. This is for the genotypic ratio, or we can say 1 to 1. What about the phenotypic ratio? For the phenotypic ratio, we will focus on the appearance. This one is axial, this one is axial, and these are terminal. So we have two of them are axial, flower, and two of them are terminal, having terminal. So we can say 1 to 1 also. So whenever you get half of the offspring recessive, at that time you should know that one of the parent will be recessive and the other parent will be heterozygous. How many different phenotypes and how many different genotype you have? You have we can say two different genotype. This is type and this is type, and also two different phenotype because this is axial and this is terminal. So whenever you get half of the offspring or 50% of the offspring are recessive, at that time you should know that one of the parents should be recessive and the other one should be heterozygous. Now, what about if the question asks about the gametes? You know that these are the gametes. For example, the question said, what's the ratio of the recessive allele in the male gamete? Let's say this is the male. And in the male gametes, what is the ratio of the recessive trait? The recessive, it's 50%. What is the ratio of the dominant one or the dominant allele? It's also 50%. Now, what is the ratio of recessive alleles in the female gamete? It's 100%. What is the ratio of dominant alleles in the female gamete? It's 0%. Okay, so these are the gametes, and maybe the question asks about the gametes and the ratio of the alleles in the gamete. What about if the question asked about what is the ratio of heterozygous offspring to homozygous? Hetero, for example, to homo, or homo to hetero. It's 1 1 because half of them are hetero and half of them are homo. So don't forget, if you get 50% of the offspring recessive and you have two unknown parents, so directly you should say that one of the parents should be recessive and the other one should be hetero. Because if you put here homo, okay, the other choice, homo with homo, at that time, this is the first case. All of the offspring will be hetero. We can't get that half of them are recessive trait. The last example that we have for here, we have the like for the example of the self-pollination that Mendel did to get F2 generation. When he crossed hetero with hetero, if you remember in the Mendel's experiment, to get F2, he did self-pollination of the parents that were all hetero. So if he cross hetero with hetero, like for example purple, P capital P small with P capital P small, or you can say hybrid with hybrid, what we can get? Let's do the parent square for it. We have P capital P small. This is for the first parent. We will form the gamete by the law of segregation. And they separate, the two factors separate during the formation of gamete. The same thing here. We have this parent and the alleles will separate during formation of gamete. Then after that, they recombine again together during the formation of offsprings. Here we have the offspring. We will distribute them in the Punnett square. The first parent will be P capital P capital. The second parent, so we have one of them, it's P capital P capital. Then we get two of them are P capital P small, and one of them is P small P small. Okay, so we can say that the genotypic ratio is 1 to 1 because one of the types is P capital, two of them are P capital P small, and one of them is P small P small. If the question asks about how many different phenotype or how many different genotype, now here how many different genotype we get? This is one, this is one, and this is one. So we get three different genotype. This is a genotype, this is a genotype, and this one. So we get three different genotype. Now we focused here in the genotype on the different letters that we get. Now let's go to talk about the different phenotype. First, we have how many different phenotype appear. First, we have this one is purple, this one is purple, and this one is also purple. And what about this one? It's white. So we get two different phenotype, either purple and also we have white. If the question about asked about the phenotypic ratio, the ratio it will be like what? We have three of them are purple and one of them is the white color. So three of them dominant and one is recessive. So we focus on the different, not letters, on the different appearance that we get, on the appearance of the organism. We have three purple to one white. Or if we say the recessive to dominant, at that time we can say 1 to 3 if we talk about the white to purple. This is the phenotypic ratio and the different type of pheno that appear when you cross hetero with hetero. And it's also named as self-pollination. When you cross something by itself, it's named as self-pollination. Also, we can say that we can get 25% are homodominant. This is homodominant. And 50% are what are P capital P small, so they are hetero. And one of them, 25%, it's homorecessive. So can we say it's equal to ratio of 2 to 2 if we talk about homo to hetero? Yes. How many homo we get? We get 50% homo in general and 50% hetero. So we can say 1 to 1. 50% homo and 50% hetero. What about the ratio of homodominant to hetero? How many homodominant we get? Only one. How many hetero? It's 2. What about homorecessive to hetero? It's also we have 1 to 2. What about if we talk about homo to homo, the ratio of homodominant to the ratio of homorecessive? So we have one homodominant and one homorecessive, so it's 1 to 1. Half of them with half of them, it's homorecessive. Now let's start to solve some questions about monohybrid cross. In a cross between two rabbits that are heterozygous for the coat color, please pay attention. The rabbit coat color, we have two types. We have black and we have the brown color. The brown color is recessive. It's always B small, B small. The black color may be B capital, B small, or B capital, B capital. Okay, so for the coat color, you should memorize it for the rabbit and also for the guinea pig coat color. The guinea pig also the black is dominant and the brown is recessive. It's B small, B small. So in the cross between two rabbits, we have two rabbits that are heterozygous. It means we have B capital B small cross with another one is B capital B small. If you know in rabbit the allele for black coat is dominant over the allele of the recessive. From here the question starts. What is the phenotypic and genotypic ratio of F1 generation? So you can do it by the Punnett square or just crossing them by that, by like that, by multiplication. If you cross them like that, you can get one of them is B capital B capital, two of them are B capital B small, and one of them is B small B small. So we can say 1 to 2 to 1. This is the genotypic ratio. What about the phenotypic ratio? Three of them are black and one of them is white, one of them is brown. This is the phenotypic ratio. Right by fraction also and percentage. If we talk about percentage, we can say 25%. It's we can say B capital B capital. Then we say 50% is B capital B small, and again 25% is B small B small. For the phenotypic, we can say it's 3 to 1, so we say 75% it's black and 25% is brown. You can do the same thing for the fraction also. For the fraction, we can say here 1 over 4 because the total number here we have 4 offspring. So 1 over 4 is B capital B capital, 2 over 4 is B capital B small, and 1 over 4 is B small B small. Okay, what about for the phenotypic? The same thing, we can say 75% it mean we have 3 over 4 for the black color and 1 over 4 is for the brown color. Now write the genotypic and phenotypic ratio.